Hi, welcome back. We are going to be learning about the cell cycle in this summary, um, which is going to include mitosis, which is mitotic division. The purpose of the cell cycle is really to describe the phases that a cell goes through as it lives its normal life. And then mitotic division is the point in which the cell needs to replicate, and so that the organism can either reproduce, um, grow, or repair any damage to tissue, tissue being the subcomponent that cells are used to produce. If you don't recall the order of structures that are inside of an organism, we can write that down here. Let me actually switch to a pencil so that we're on the same page here. So it can go down here again, and it's going to be organelle, which are used to make cells, which are used to make tissue, which are used to make organs, which are used to make organ systems, which are used to make organisms. Okay. Even before that, you can go with molecules right here, right? So molecules. Mhm. And you can say atoms before that. After organisms would be populations. So these are called the levels of organization. So you should know this by now, but we're going to continue to add to this as we go through the year. Levels of organization. So let's start off. So we're in the levels of organization. We are now talking about the cell and living the life of a cell. So we're going to start off. The most important portion of a cell's life is called interphase. The cell spends most of its life in interphase, as it is generally how it's living. G1 is currently considered the part where the cell grows. G, all the G's are grow. That's what G stands for, growth phase. So G means grow, but in grow 1, the cell is conducting its normal life. Sometimes people consider G1 to be separate from G0, in which G0 will be the normal life. We're just going to consider it both the same, and so G1 will be where it grows and lives its normal life. Why is it growing? It's growing after cytoplasm, or I'm sorry, cytokinesis. Cytokinesis was where the splitting of the cell happens, and so at that split, it's kind of half the size it used to be, and so it grows to the size it needs to be in order to complete its function. Then we have S phase, which is where the genetic material is replicated. So S phase is considered synthesis phase. And remember, synthesis means to make. So synthesis, what are you making? You're making a copy strand of DNA. And then G2 is where the cell grows once again. So we have growth 2. But this time it's growing to get ready for the split. So it's going to get bigger in preparation of going through mitosis. Okay. Now it can only go to a certain size. Cells have a maximum growth limit before the amount of energy required to send things through that cell becomes too much. If you can imagine, we had a cell like this. The time it will take to get materials through that cell would be much, much longer than if I had one cell here and one cell here, right? Each cell can go through that process of sending materials through the cell a lot quicker than having one size, right? So the area of the cell is the same. However, each cell can do that process faster than that one singular big cell. And so that's why multicellular organisms are made of more than one cell rather than relying on one cell. It's because it becomes really inefficient when they get too big. So that's the interphase. Growth phase 1, synthesis, and the growth phase 2. If a cell gets into growth phase 2, it's going to check to see if it's ready for mitosis. If it's not ready for mitosis, it's either going to go back, get all the materials it needs, or if the mistake is too big, it's just going to lyse, which means it's going to pop. It's going to kind of self-destruct in order to avoid replicating any issues that that cell has and passing it down to future cells, because that will continue to replicate, and that's a big problem. So it will actually just kill itself to avoid that. But it's cool. Anyway, so then we go to mitosis, which is mitotic division. The easiest way to remember this is PMAT, PMAT. That is the order of events for mitosis. In this test, you will be asked to look at images and know which phase is going on, and also kind of understanding the processes. So this is really important, more so important than the interphase. So you need to know the phases of mitosis. So during prophase, what's essentially happening is that the chromatids actually are going to form. So when you get those X-shaped chromosomes, remember that those are chromatids. And they're going to form during prophase. During metaphase, those chromatids align in the center, and the spindles attach to each side, getting ready to pull those chromatids apart. In anaphase, they are actually pulled apart, so you'll start to see them kind of start to pull away. And the entire purpose of anaphase is to ensure that each cell ends up with the same amount of chromosomes. So it pulls them apart equally, right down the middle, so that each cell that is then formed from mitosis ends up exactly the same. That is the key of anaphase, ending up with the same amount of chromosomes. And then telophase is once those cells are really pulled apart, everything starts to reform. So the nucleus will start to reform, the organelles will start to come back, the cell nucleus will come back, and the division will start to happen. Of course, it doesn't actually start to divide, because cytokinesis is that actual division. So this is not a part of mitosis. It's simply the step that's needed to complete mitosis. So cytokinesis will actually pinch off the cell membrane, and the cytoplasm will divide into two different cells. This entire process is a form of asexual reproduction. This A means non or counter. So it's a counter to sexual. Sexual being two organisms, asexual meaning one organism creating a clone. And that is the key here. So you're creating a genetic clone through this process. And it's used mostly to either grow an organism or repair tissue, or to just replicate if you're a single-celled organism, pass on your genetic information. All right, so that is mitosis, growth, synthesis, and growth for interphase. And then mitosis, you got PMAT, prophase, metaphase, anaphase, and telophase going on here. Again, PMAT going to be the most important piece. Really focus in on what is happening in each of those phases, as that will definitely be on the test I'm telling you now. All right, see you later. If you have any questions, feel free to ask.